are doing, commercial is also doing this analysis. This is for databases for the persons who work on natural products. They found a good product, they say our product is new one. So now you have to check this databases, put some data and come to know whether sample is known or earlier some people have worked or it's a new one. This is the what Madam Bharti was telling about the artificial intelligence. So we have started here. So next time when you go for diabetics or anything after 30 or 20 years, they take your picture, eye picture only. And they will tell you whether you have diabetes or not, you are not diabetes, how much, what level diabetes or cardiovascular disease, everything. So this is what Madam was telling morning, AI, artificial intelligence. So the many ways is coming for that. So no, in future you will need a doctor need your picture only. The picture he will analyze and he will tell you have this disease, you looks like this. So microbiology. So we have power of bacteria in human body. So we have many bacteria in our body, and they regulate everything. So this is type. You can see the human microbiome. Plant also have a bacteria. Lot of bacteria. Plant nitrogen fixation, soil, other things in agriculture also. So this is the all microbes. So this is the observation of the century because gut bacteria. So it's a gut bacteria, our stomach bacteria, they are responsible for all diseases. Very quickly, you have population shift, then we have a disease. You can understand more this. The lean, the thin lady, they have this type of bacteria. And fatty lady, they have this type of bacteria. So when you develop the food product, look for the vegetables which having this type of bacteria or this type of bacteria. So you can develop the food for the fatty people or thin people. So what is the natural sources? What is the sources? God gives lot of things. Let us find out that because from God we also go there. Sometimes we go Himalaya somewhere, look for the God who help you, who understand you, who give you some wishes. But here microbe also, you go to the good search of good microbes. Where you get the good microbes, so where you can get, let us see. So this is aloe vera. So here we are working on the aloe vera cream is very good for the skin. As I told you, I am working on the skin. So we are looking for the microbes inside that, produce the gel. This is important. Let us see, this is a slide. From childhood we are looking the slide. The termites comes and eat the wood. But why it is coming? So what is the reason behind that? Anybody idea?
The gut microflora.
Gut microflora. So it contains not gut microflora, something else. This wood contains cellulose, and this contains the cellulose enzyme present in the termite gut.
Sir, some microbacterium is responsible for these enzymes.
We are okay. So we are our aim was for the cellulose enzyme. So we found the cellulose enzyme in termite gut. This is a joke. Let us see. You will be happy to tell them. David is active his whole life, died in 12 years, but after sleep mostly survived to 100 years. Is true or false?
Very true.
Very true. So no need to wake up early in the morning and go for exercise. But there is something else from as a scientist or microbiologist things. What message? So tortoise have longer life because slow metabolic rate. So we are working on the gut of this microflora for this tortoise. So let us see what enzyme, what chemicals we get which reduce your age. So increase your age and reduce your so this is we are looking for the angle for what God is giving. So we are looking for the source from there. So this is also somewhere you can get the microbes, some good microbes. So this is the what nosy. We throw the nosy and throw it. You understand? From nosy, we got this bacteria, very good bacteria, powerful bacteria is there. So they kill many bacteria, antibiotic substance from nose. So this is another bacteria. So this is some scientifically the proline, PP. So this is from here we can, with the help of chemist, we can synthesize new type of the protein compounds. So they found from the, because normally fish and the other crabs, they don't have a less skin diseases. So we also working on our lab is also working on vitiligo. So vitiligo is a good example of we work for that buffalo patches buffalo, white and black buffalo is the good model for the study for the vitiligo in human being. And the pig skin is very much close to the human skin, good model to study the bone healing and diabetic disease. So our skin and pig skin are very much close. So this is some other microbiome we have studied. I found some compound from the potatoes which reduce the blood pressure. Helps to growth in the gut bacteria, ugamine, a new compound from potato, low blood pressure. This is a watermelon from microbes. So this is something like Viagra, sildenafil from different varieties. So this is not important now. Little bit sometimes my student Vipul is here. He is working on acne. So we have a different population of acne. So what we did? Quickly I will go. So we did lot of analysis, and one analysis we did for the Najak also, MLST analysis. So different type of antibiotics. So I will come. So this is all skin microbiome. How much skin, what is the method, other things you can get. So this is not interested for you, but a little bit. Wait. This is our work. So the person is suffering for the acne. So doctor give lot of treatments, but he could not treat the drug. So could not get it from the acne. So he spent lot of money. Then doctor sent the sample to us. Then we did some analysis and we give them. We asked to combine this antibiotic, give to them. So after some time, he was perfectly cured. So this is paper is also published in the journal. So this is some real molecular analysis which may not be useful for you if I will go. So last time. So this is all molecular analysis, how we are doing the microbial population of different skin samples or acne samples. So this is all for who is understanding bioinformatics. People, they can understand what is this. So this is the summary. So we have a different type of subtypes. This is a different type of subtypes. After all molecular analysis by our we found the different type 1, 2, type 3, type 4.
Informative talk, and I think everybody enjoyed. If any questions do you have, then you may ask one.
One thing I ask you by the student Vipul, he got about that half thesis we have sold to the industry. He got about the 10 lakh rupees from the industry for his work.
Now trying to come to see some about in general biotechnology of disease, especially non-communicable disease like cancer and others, and how its solution and latest solution, what will be the latest solution, and then that's indicating towards opening new industry. That is my goal. So my vision is like to develop a novel technology, especially for early diagnosis is important. So diagnosis of early diagnosis of any disease will help to. So development of new tool for diagnostic tool is one of my mission to betterment of the common people, and also to address socio-economic. That discovery will help directly to social people how it will be beneficiary. So then these are all my mission in general. Like what the theme is, like two in major field I call personally is medical biotechnology and marine biotechnology. So before this kind of like so objective is to develop, as I mentioned, develop a very low level expression of mRNA, for example, for based on diagnostic tool, and also clean like various disease. You can make a novel diagnostic tool based on case control study like coronary heart disease, and another objective is to develop eco-friendly herbal medicine, which will be the leads for industry development. Like for example, for this to fulfill this objective, there is that for any basic scientist people can understand. Based on a synthetic plasmid, you just make a synthetic DNA and cloning a plasmid to make a synthetic plasmid, and that will help us enormous to diagnose many diseases. Like for example, as I meant, I have developed this kind of plasmid which will help you to estimate quantitatively in terms of like a femtogram of expressed mRNA of any. And as we know, like expression of upregulation of downregulation of various cytokines, immunologically important things, are playing major role for many diseases. Like for example, inflammation, anti-inflammation. These are two tug of war, and these are all upregulation. So if you have a sophisticated tool for measuring this kind of mRNA, then you can develop a diagnostic tool just taking a drop of blood and then measure all those within an hour, and then you can tell what kind of disease you have, either any non-communicable disease could have or not. So this technology could be easily developed based on synthetic DNA. And similarly, we have done also, here we have already developed one tool, we can measure 18 different that type of cytokines. And similarly, we have another approach like case control study. One group of people with a patient, another group of is non-patient, non-male. And based on that, measuring all bioparameters from patient group versus non-patient group, then all data will be analyzed by different statistical methods, and make a, like for example, univariate analysis of that disease makes one APOE is the not significant, all are significant. Gradually, when you discard univariate to multivariate logistic regression analysis out of so many parameters, and you can predict 5 different parameters are highly significant statistically, then we made a formula, and then we can, now we having this formula, we can take a drop of blood, measure all these 5 parameters and ask them whether you are under risk of that disease or not ahead of time. So that way we can develop a tool, and then our goal is to, after making that, and similarly another parameter, these are biochemical parameters, and these are all immunological parameters, and these are like a immunological, and then that, and many genetic factors also, that way you can measure. So based on overall you can make, and then we publish all those data, and then we found blood group also responsible, and then that way we publish lot of paper, but based on our goal would be to make a biometric chip, and from biochemical, immunological, and genetic parameters. All those parameters we can measure for a specific disease, and we can develop a biometric chip so that any person will come and take a drop of blood, like pinching with needle on finger, and then that measure all those, and then chip will tell you like whether you are disease prone or not. So that kind of technology can coming up from this analysis. And then another important disease I like to mention regarding white spot disease in shrimp aquaculture industry is really drastically destroying this industry, and loss of huge crore money extraction each year due to this disease, white spot disease. And then there is no solution. White spot carapace and shrimps are dying quickly within 3 days. Enter bonds, which is 99% individual dying due to disease. Such economic destruction going on. There is no solution. So there is another approach we like to highlight you, to inform you. And then, for example, if the idea came to my mind when I went to talk with farmers, and they said 1% to 10% are surviving even in the affected wards. My question, my scientific mind asks immediately why they are surviving. It is showing infectious disease, why they are surviving from this disease. So if we could have a technique, so identify those parents, so that then we could develop a disease instead of theory, and we can avoid the disease based on the techniques. We can screen individual parents and let them breed and get the seeds and given to farmers will be avoided and rectified. That is called disease resistant species could be developed from that. And yes, we have developed that type of DNA-based technique, and that has been patented by 7 countries, including Malaysia, Singapore, Malaysia, Indonesia, and Vietnam, and as well as India. All 7 countries have been patented this technology, and it is supported by the DBT government of India. And then we have tested all those markers, whether it will be applicable practically or not. With good stock means parents would be, hatchery will be tested, and we have done in the laboratory scale marine aquarium. Yes, it is possible if we select individual based on DNA markers, and then we inject individuals, and then we have seen that survivability as well as these are survivability versus mortality among resistant and susceptible, and as all are significant, statistically significant, and copy number of virus also increase in susceptible, and which is not going increase and resistance, although we have injected. So these are data has been statistically significant, and now it should be applied to the marine hatchery, which can produce really disease seeds. This type of techniques will be able to cure, avoid the same disease. And we so far developed this type of marker, like the 80 different markers, all are statistically significant and also tested in marine aquarium in the laboratory condition. And yes, we can select individual. Now our scientific goal from this program to identify new genes which are linked towards disease and which is novel. In shrimp system is shrimp in Malaysia and other places also shrimp industry is very important, especially marine shrimp. And these kind of things will help really a lot. And that way, then we have tested all those, even where at least at this moment, wild food stock is used for cultivation of seed culture. And we have tested entire Indian coastal area. We have found a couple of places like this place, this place, this place, and this place are the safest place to collect food stock for breeding program and immediately. But at this moment, there is no such technique. We can call SPR, specific pathogen resistant for seeds generation, nowhere. So we would be, this marker, DNA marker would be first time helpful to generate disease resistant seeds. This is one program, very interesting program. And we have done all publication from this program. And now, these are all publications, and a lot of sequence has been, these are patent also, patent in different countries. And then a lot of DNA sequence has been submitted, and a lot of work is going on in this field. These are patent from Singapore, Malaysia, Philippines, Indonesia, Vietnam, Thailand, India. Shrimp industry. That will be really helpful in Malaysia as well as entire this Asian country. This is very good, important. This disease is very important, and is a loss, destroying crore money from this industry. No revenue. And this type of techniques will help really a lot to prevent this economic destruction. And so the last one is my medical vital program, as I mentioned regarding the development of better and safer medicine to combat non-communicable disease. For example, cancer, using natural resources. As you know, India has huge natural resources, not only India, Malaysia also. Yesterday I have seen huge resources available. We can encash this resource to develop a better, safer. Why call better and safer? Means, as you know, for cancer, for example, there is a true drawback. One is huge side effect chemotherapy. has huge side effects. That is the only chemotherapy is the solution. Other radiotherapy and operation all are management, but only therapy is chemotherapy, and there is no other solution. But that drawback, major drawback of chemotherapy is huge side effects, and also drug resistance appearance is another problem. So they are making cancer cells are making resistance in a specific drug. So then that drug will not work anymore. Although it's so expensive. These are three major drawbacks. A huge expensive and also drug resistance. Another is a huge side effect. This is the main problem in cancer. And even if you have detected earlier, that you cannot control. Ultimately people are dying. This is the most helpless situation of cancer. And that I think I have done so many work in this field also. If you see there is some solution and hopeful we are. That's why it will basically each and every program which are so far described, and I will establish also, will be evolve a new industry for each and every technology. So that's my goal is. Like for example, this is the basic things I know from basic science, that is cancer, for cancer, then for controlling cancer or any non-communicable.
First of all, I want to thank you all for being patient and waiting for me, and I'm sorry for being late for this session. Okay, the topic for today for me is the emergence of quinolone multi-drug resistant Salmonella Kentucky from zoonotic foodborne environment in Tunisia. This work, first of all, I have to give you the idea. This is a small work from a big project we started at Pasteur Institute of Tunis. The budget was from the network Pasteur Paris. It's called the Epidemiological and Molecular Survey for Disease Risk Assessment of Multi-Drug Resistant Salmonella in Tunisia. The teamwork, of course, Pasteur Tunis. Also, we have Frederick Loeffler Institute in Germany, and also Professor Gatom, which help us in genotyping. The project objective, the main objective is to establish and build an appropriate effective database which will serve to monitor and better detect Salmonella infection in Tunisia, to detect rapidly epidemic outbreaks and emergence of multi-drug resistant of Salmonella from different sources and geographical region in Tunisia also. Although, after the main objective, this is a specific objective, just to, as Dr. Gatom, I don't have time now. Okay, this work started with 1,404 isolate zoonotic human and environment. We included 2 serovar, Kentucky and Tefimurium, because in Tunisia, this is considered the top serotype in Tunisia during the years from 2010 to 2014. The project ended with 4 manuscripts: emergence of Salmonella multi-drug resistant in Tunisia, quinolone, beta-lactamase, and integron. The manuscript already finished, and we are submitted for publication, but we still having genomic island and whole genome sequencing with the German institute that's still working on it. Excuse me, I will talk about only quinolone. I'm not going to mention other work because it needs more time for me. For the quinolone. For the quinolone, we have 2 main objectives. First, monitor recent trends and emergence of nalidixic acid resistant and Salmonella Kentucky Tefimurium in a collection from 2010 to 2014, and investigate the genotyping diversity between different strains isolated from different regions and different sources. We used the period where from 2010 to 2014. During these years, we have a total of 10,098 Salmonella strains isolated during these 5 years, but we concentrated only in Kentucky and Tefimurium because it's a huge, and by the way, this work is done by 3 students. For the quinolone, a master's degree student, she finished it last year, this one. Okay, we used antimicrobial test for 23 antibiotic, amplification by PCR, sequencing, pulse field gel electrophoresis, and multi-locus sequence timing. All these at Pasteur, Tunis. MLST done by the student in the German Institute. The reading of the sequencing by the student of Professor Gautam, what's his name? Where is he? Yeah, he did the reading for us for the MLST. Okay. For the antimicrobial susceptibility test, we use beta-lactamine, quinolone, aminoacid, macrolide, sulfonamide, phenol, and tetracycline. But this works only for quinolone. We detect quinolone-resistant determinant region, for that is chromosomic, and also the plasmatic mediated quinolone resistant. From the isolates, the amplification of seven genes, presence of PARP and GIR, that is chromosomic, and quinolone A, B, and S, which is plasmid. Also for the integron GIP A and AS. The results we obtained. During these years, Kentucky and Tefimerium and other. The Kentucky and Tefimerium, Kentucky represent 10.48, Tefimerium represent 3.4, other serovar will represent 86.09. For Salmonella Kentucky, oh sorry. Yeah. For Salmonella Kentucky, there is variation in the isolation year during the years, but we have to concentrate that during 2011, the isolation rate is low. This is not because of the low isolation of the Salmonella law, but this is during the revolution in Tunisia. So isolation and the research stopped during this year. I'm sorry because this is in French.
Sometimes it's a big population, sometimes less population.
No, but in 2011 this is the first population, for the first revolution in Tunisia. So it's a revolution. No one is working. So for the distribution for quinolone, I'm sorry. For distribution for quinolone, most of it from the poultry, Wallahi, that is poultry, 82.8, and after that it will be the bovin, that is from, how can you, meat, I'm sorry because it's in French, from meat, and others from the environment. For the Kentucky, the most isolation, the maximum, it was from the animal, then from the environment. Tefimerium was from the human and from the animal. Animal, that's the food, sorry. Okay. The most region that isolated the quinolone resistant was from the capital, Tunis, and the shore, we call it Sahel region, that is near the sea. Okay, for the antibiotic resistant. For the antibiotic resistant, high resistance for nalidixic acid, ciprofloxacin, and cipro. Also here for amoxicillin and for AMC, ampicillin, and ticapsulin. Others, variation. Multi-drug resistant represent 66.6 for Kentucky, 26.8 for Tefimerium. For the PARP and GIR, as I said, quinolone mediated chromosome. So quinolone resistant must be founded. So all positive. The problem in the plasmatic, because Salmonella, it's rarely identified the quinolone S or B or S. It's really rare. So here we found only in two strains, the positive was for quinolone B. Okay, these two strains was Tefimerium, two strains, Tefimerium isolated from human asymptomatic, okay, and the quinolone B by sequencing identified as quinolone B19. For the AAC also observed in one Salmonella Tefimerium in association with quinolone B and GIR and PARP. the sequencing of GIR of these two isolate that there is a mutation in a specific region. So the quinolone resistant, and this mainly mediated by mutation in the level of DNA G-base. Multi-drug resistance, MLSD and pulse field gel electrophoresis give us two different profiles for these two unique, or that having the plasmid quinolone resistance. And for the MLSD, we found it ST34, ST313. The most important thing in this point, that we started with 145, 104 only phenotypically resistant to quinolone, 4 strains, not 4 strains, 2 strains, not 4, 2 strains of Salmonella typhimurium genotypically resistant to quinolone. Okay, the most important things in this study, that this is the first report of Salmonella typhimurium ST34 in Africa, and also the first report of quinolone B19 in Tunisia. That's the most, and of course this one is in favor of the student who give us these results for this one. Difficulties, scientifically we don't have any problem during the project. Financially the budget was not enough to cover the first part due to huge number, because we covered 10,000 Salmonella strains. It's a huge number, so the budget was not enough. And impact perspective, we are now working with this institute in Germany to have to cover different approach in Salmonella infection and multi-drug resistance. This is my team, okay? This is the student, she's worked in the beta-lactamine, I didn't represent it here. Now she's in Germany in the institute finishing her PhD work. This is Nadine, who finished the quinolone in her master degree, that I represent her work now. And now she also in Germany studying, also finishing her PhD in multi-drug resistant from a plant extract. And this is the smallest one. She finished in December her master's degree. Also she took part, not for Salmonella, for the E. coli enterohemorrhagic strain. And this is my team. I'm proud so much for them. And merci, thank you very much.
I'll show you why we design how we design. And one is antagonist. So these two term agonist means the molecules which have affinity to bind with a particular receptor, because all the drug molecules which we are designing, they are for, whenever you design a drug, there are problems with that. No doctor says